The Department of Agriculture, Land Reform and Rural Development at DALRRD Evaluation Center established the National Gene Bank in 1998. Samples of seed and vegetative species are mainly obtained from communities farming under traditional farming methods to produce their food, and in some instances using their indigenous agricultural knowledge and practicing their cultural beliefs. Some of the crops collected are mung bean, calabash, pumpkin, sorghum, maize, and cowpeas. When material arrive at the gene bank, samples are processed to determine their health and quantities. Currently, there are about 6,300 samples of seed and vegetative species of crops, medicinal plants, and some wild plant species. Wild plant species include some crop wild relatives. After processing, seed samples are sealed in aluminum foil bags to prevent them from interacting with the outside environment. The criteria for a sample to be placed in storage is that the seed must have a seed moisture content of between 4 and 7% and a germination percentage of 85%, and with the seed quantities of a minimum of 1,500. Packaging is done to allow for monitoring of seed viability during storage by using a bulk bag and 4 smaller bags to allow for extraction for monitoring. It is important to clean equipment and other objects that are going to be used during subculturing, as well as for the scientist or technician to maintain cleanliness throughout the process. The gene bank maintains some vegetative samples that cannot be stored as seed. These are prepared in the laboratory to store buds or shoots in tissue culture using appropriate media culture solution. This is done for taro, sweet potato, and cassava. A sterile environment is necessary to prevent contamination and loss of sample during storage. As explants grow during storage, regular subculturing is performed as required to keep them alive from dying out. Test tubes must be properly closed during storage to avoid explants to be contaminated. The gene bank uses a walk-in storage facility for active collection to allow for access to provide material for utilization. Sweet potato sample stored in tissue culture are duplicated in the field gene bank. Protocols are to be investigated to duplicate medicinal plants stored in the field in tissue culture. When placing processed samples in freezers for long-term storage, samples are packed in 4 small bags for extraction every 5 years attached to a bulk bag. A duplicate sample in the National Gene Bank is deposited with the SADC Plant Genetic Resources Center in Lusaka for safekeeping. The bags are placed in cartons to allow for proper tracking and of the sample for easy location when needed for extraction. The freezers take about 100 cartons. A monitor is attached to the freezer to record daily temperature and relative humidity. The room holding freezers is monitored daily to maintain temperature. Data generated for each accession is documented on paper and the SADC Documentation Information System developed by the SADC Plant Genetic Resources Center. The SDIS is separated into modules for registration, passport data, and samples in storage. Data recorded in the SDIS can be retrieved to create reports such as characterized samples or samples due for regeneration. Additionally, the National Gene Bank reports the samples it holds to the FAO early warning system. The total complement of the gene bank is 8.